Welcome everyone. In part 4 of this chapter, Biosignaling, we will discuss three different topics. In the first topic, we will understand how voltage-gated ion channels contribute to electrical signaling in excitable cells, such as neurons. In the second topic, we will discuss how nuclear receptors regulate transcription factors in the nucleus. In the third topic, we will understand cell cycle regulation mediated by protein kinases. Certain cells in multicellular organisms are excitable, which means that these cells can detect an external signal and convert them into an electrical signal and pass it on to other cells. Now, this electrical signal could arise because of a change in membrane potential. Now, excitable cells, such as nerve cells that are shown here, play central roles in nerve conduction, muscle contraction, hormone secretion, sensory process, learning and memory, and so on. The excitability of sensory cells, neurons, and myocytes depend on ion channels. Ion channels, as you may know, are central to transporting various different inorganic ions, such as sodium, potassium, calcium, or chloride ions across the plasma membrane in response to various stimuli. Now, these ion channels can be open, closed, or gated, and they can be activated by secondary messengers like CAMP or other inorganic ions bound to them. So the function of gated ion channels is to regulate transport of ions across cell membranes. Specifically, these gated ion channels respond to changes in the membrane potential or ligand binding to specific receptor sites. There are many important roles in the nervous system for these gated ion channels. For example, voltage-gated sodium channels are important in neuronal signaling. Membranes are electrically polarized. The inside of the cell is typically negatively charged compared with the outside. A Vm of minus 50 to minus 70 millivolts is maintained within the cell. Vm means electrical potential. The membrane potential is largely due to asymmetric transport of cations by the sodium-potassium ATPase. Now, this particular transporter transports 3 sodium out of the cell while taking 2 potassium inside the cell. Flow of ionic species across the membrane depends on its concentration gradient and overall electrical potential. Shown here is a sodium-potassium ATPase that is embedded on a cell membrane. This transporter produces a transmembrane potential of about minus 50 to minus 70 millivolts by sending out 3 sodium ions out of the cell and bringing in 2 potassium ions into the cell. The direction of spontaneous ion flow across a polarized membrane is dictated by the electrochemical potential of that ion across the membrane, which has 2 components. The first one is the difference in concentration of the ion on 2 sides of the membrane, and the second is the difference in electrical potential that is typically expressed in millivolts. Given that the ion concentration difference and an electrical potential of about minus 60 millivolts is inside the cell, if a sodium or a calcium ion channel opens, it will result in a spontaneous inward flow of sodium or calcium. And this results in depolarization. Whereas opening of a potassium channel will result in a spontaneous outflow of potassium from inside to outside. And this causes a hyperpolarization. In this case, potassium moves out of the cell against the electrical gradient because the large concentration difference exerts a stronger effect than the electrical potential. So a high concentration of sodium inside and low outside pushes potassium outside, and that movement is basically going from a high concentration to low concentration gradient. And so that particular gradient is higher than the electrical gradient. For chloride ion, the membrane potential predominates. So when a chloride ion channel opens, chloride ion flows outside. So it is the electrical potential gradient that predominates for a chloride ion, whereas a concentration gradient predominates for a potassium ion channel when it is open. And this happens in tandem with the sodium-potassium ATPase. Signaling in the nervous system is accomplished by networks of neurons, as shown here. Neurons are specialized cells that carry an electrical impulse, as shown here, from one end of the cell, called as the cell body, through an elongated cytoplasmic extension, called as an axon, to the end of the cell, called as a synapse. Now the electrical signals trigger release of neurotransmitter molecules at synapse, carrying the signal to the next cell in the circuit. Now the cell that is interacting with the first neuron is called as the postsynaptic cell. And the cell that initiates the signaling is the presynaptic cell. Now, signaling in neurons is accomplished via ion channels. There are three different kinds of voltage-gated ion channels in neurons: voltage-gated sodium channels, voltage-gated potassium channels, and voltage-gated calcium channels. Now these three channels work in tandem to maintain an action potential, or the electric impulse that is shown here. So how is this action potential maintained? Let us discuss as to how this is accomplished. Shown here is a zoomed in view of the axon of a presynaptic neuron. Initially, the plasma membrane of the presynaptic neuron is polarized, which means that the inside is negative and the outside is positive. And this is achieved through the action of sodium-potassium ATPase, which maintains a membrane potential by transporting 3 sodium ions out and taking in 2 potassium ions. When the cell body of this neuron receives a stimulus, the information is passed along the axon as an action potential, and this could be in the form of a neurotransmitter. Now, this neurotransmitter could initiate the action potential via activating a voltage-gated sodium channel. Now the voltage-gated sodium channel, when opens, sodium is rushed in to the cell for a split second. Now the influx of sodium through the sodium channels depolarizes the membrane locally, and that's what's shown here as depolarization. Now, the sodium channel only opens for a split second and then closes. And as a result of this depolarization, the potassium channel that is next to the sodium channel, that sodium and potassium channels sort of line along the axons in neurons. Potassium channels open up so as to repolarize the membrane. Now the resulting potassium efflux repolarizes the membrane locally and establishes a negative membrane potential. Now this depolarization wave is initiated locally, and once this wave of depolarization reaches the end of the axon, it activates calcium gated channels or voltage gated calcium channels, which open, resulting in the rushing in of calcium. Once calcium comes into the cell, it acts as a secondary messenger. And what it does is it triggers a release of acetylcholine by exocytosis to the synaptic cleft. And that's what's shown here. And this is achieved by the action of increased calcium concentration. Acetylcholine diffuses right into the synaptic cleft as shown here, and it comes out of these vesicles from the presynaptic neurons. Once in the synaptic cleft, as you can see, and the image shown on the right here, these acetylcholine molecules enters the next neuron. And when it enters the next neuron, what happens is these neurons again initiate an action potential. And this is the cell body of the postsynaptic neuron as shown here. And when acetylcholine enters via acetylcholine receptors, another action potential is triggered. Thus, the message is passed to the next cell in the circuit. We see then that gated ion channels convey signals in either of two ways: by changing the cytoplasmic concentration of an ion, such as calcium, which then serves as an intracellular secondary messenger to initiate acetylcholine release, or the cell changes the electrical potential, or VM, and affects other membrane proteins that are sensitive to the electrical potential. Now, the passage of an electrical signal through one neuron and onto the next neuron illustrates that a change in chemical concentration, or the concentration of the ions, and the electrochemical gradient are both at play here for signal transduction, and especially in case of neuronal signaling. Let us move on to the next topic: nuclear receptors. Let us examine as to how transcription is regulated by nuclear hormone receptors. Now, we discussed certain hormones and hormone receptors like insulin and insulin receptors. Now, there are certain hormones like steroids, retinoic acid, and thyroid hormones that are different from hormones that we discussed. The difference is in their mechanism of interaction with their receptors. For example, these hormones bind directly to their receptor in the nucleus to alter gene expression. Steroid hormones such as estrogen, progesterone, and cortisol are too hydrophobic to dissolve readily in the blood and are transported on specific carrier proteins from their point of release to their target tissues. As shown here, a steroid hormone binds to serum binding protein, which takes it to the target tissue. Once the target tissue is arrived, serum binding proteins diffuse away, and the hormone, because they are hydrophobic, diffuse through the plasma membrane and through the nucleus and binds to specific receptor proteins as shown here. Once the hormone binds to its receptor, it forms homo or heterodimers with other hormone receptors. So it basically dimerizes the receptors. And these dimers and tetramers bind to specific regulatory regions that is shown here, and the abbreviation is HRE, or hormone response elements. And this is in the DNA adjacent to specific genes. As a result of this, the receptor attracts co-activator or co-repressor proteins, and with them regulates transcription of the adjacent gene, as shown here. And this either results in increasing or decreasing formation of mRNA as a result of transcription. So this basically means that the hormones that bind to the hormone response elements could either increase or decrease mRNA formation. So either upregulate or downregulate transcription. Now this altered levels of hormone-regulated gene product produce the cellular response to the hormone. So mRNA, if increased, results in translation of new proteins that leads to altered cell function. Now, if the mRNA is downregulated or not produced as much as the cell usually produces, then translation of this mRNA is decreased, and as a result, the new protein formation goes down. That also affects cell function. So this is how nuclear receptors regulate transcription of certain genes, and as a result, translation of certain proteins. Let us discuss the next topic, cell cycle. One of the most dramatic manifestations of signaling pathways is the regulation of the eukaryotic cell cycle. Cell division occurs in virtually every tissue. A cell's decision to divide or not is of crucial importance to the organism. Also, proper cell division requires a precisely ordered sequence of biochemical events. Cell division accompanying mitosis in eukaryotes occurs in 4 well-defined stages. In the S phase, or the synthesis phase, the DNA is replicated to produce copies for both daughter cells. So DNA synthesis happens in the S phase. In the G2 phase, new proteins are synthesized and cell approximately doubles in size. G indicates gap between division. In the M phase, or mitosis phase, the maternal nuclear envelope breaks down. And if you zoom into the mitosis phase, you can see that it is divided into 4 different phases: the prophase, metaphase, anaphase, and telophase. Now, in prophase, the maternal nuclear envelope breaks down. In the metaphase, the paired chromosomes are pulled to opposite poles of the cell. In the anaphase, each set of daughter chromosomes is surrounded by a newly formed nuclear envelope. And in telophase, cytokinesis happens and it pinches the cell in half, producing 2 daughter cells. After the M phase, what happens is the cell goes directly to G1 phase in case of embryonic or rapidly proliferating tissues. After passing through mitosis and into G1 phase, a cell either continues through another division or ceases to divide, enter a quiescent phase that may last hours, days, or lifetime of the cell. This is called as a G0 phase, which is a terminally differentiated phase when cells withdraw from the cell cycle indefinitely. Now, the re-entry point is when the G0 the G0 phase goes to G1 phase, where cells enter into G1 phase after going to G0 phase. And when a G0 phase initiates, a cell begins to divide again. It re-enters this division through G1 phase. And G1 phase is when RNA and protein synthesis happens. No DNA synthesis happens at this stage. Now, stem cells retain the potential to divide and to differentiate into any number of cell types. Cell cycle is regulated intracellularly by cyclin-dependent protein kinases. Cyclins are proteins that control cell cycle. Cyclins bind to a family of kinases called cyclin-dependent protein kinases, or CDKs, to initiate cell division. 10 different cyclins and 8 different cyclin-dependent kinases are present. Certain animal cells undergoing synchronous cell division, some cyclin-dependent protein kinase activities show striking oscillations. These oscillations are the result of 4 mechanisms for regulating cyclin-dependent protein kinase activities. First, the cyclin-dependent kinase phosphorylation or dephosphorylation. Second, controlled degradation of cyclin subunit. Third, periodic synthesis of cyclin-dependent kinases and cyclins. And last but not least, action of cyclin-dependent kinase inhibiting proteins. The precise timed activation and inactivation of a series of cyclin-dependent kinases produce signals serving as a master clock that orchestrates the events in normal cell division and ensures that one stage is completed before the next begins. An example is shown here in this plot of kinase activity versus time. In this case, cyclin E CDK2 complex activity peaks near the G1 phase and at the boundary of the S phase. When the active enzyme triggers synthesis of enzymes required for DNA synthesis, and that's when this particular complex activity is at the highest. Cyclin A CDK2 complex, on the other hand, have an increased activity during the S and G2 phases. As you can see, it sort of rises during the S phase and sort of saturates at the G2 phase. Cyclin D is active as long as growth factor is present. Now, cyclin B CDK1 activity, as shown, cyclin B CDK1 activity peaks at the M phase and sort of drops down at the same phase as well. So, depending on which cyclin and which cyclin-dependent kinase is acting, the activity peaks at a specific phase and drops down at another phase. Cyclin-dependent kinases, or CDKs, are active only when associated with a cyclin. The crystal structure of cyclin-dependent kinase CDK2 with and without a cyclin reveals the basis for the activation of this enzyme. Now, without cyclin, the structure is shown here. In this case, CDK2 folds so that one segment of the T loop that is shown in red obstructs the binding site for protein substrates, which means that when the T loop is lying like this, no substrate can bind to CDK2, and CDK2 is inactive. Now, there are also other residues that are shown here. One of them is tyrosine 15, which is phosphorylated when CDK2 is inactive. And as a result of that, a negative charge is occupying this region where ATP should bind. Now, only when tyrosine 15 is dephosphorylated can ATP come and bind. In this structure, ATP is sort of docked in the site to represent a nucleotide binding site. When cyclin binds, now this is the cyclin subunit that binds CDK2. When cyclin binds, it forces conformational changes that move the T loop away from the active site and reorient an amino terminal helix, and that helix is shown in green, which results in bringing a residue critical to the catalysis into the active site, and that residue is glutamate 51. So when cyclin binds, the T loop moves away from the protein binding site, and that brings this green helix towards the active site along with glutamate 51. Now, if you consider another structure of CDK2 going from inactive to active, you can see that once GLU51 is in the active site, a threonine residue in the T loop gets phosphorylated, and its negative charges are stabilized by interaction with 3 arginine residues as shown here. If we were to zoom into this particular part of the active site, we will see that the phosphorylated threonine residue interacts with arginine residues that are positively charged. Now, CDK2 gets active or becomes active as a result of dephosphorylation of tyrosine 15. And as a result, ATP can bind in the active site and CDK2 can initiate phosphorylation of its substrates. Cyclin-dependent kinases are regulated by phosphorylation and proteolysis. As a cell enters mitosis, the M-phase cyclin-dependent kinase is inactive. As cyclin is synthesized, as shown here, the cyclin-dependent kinase forms, and as you can see, this complex is inactive. That is because the T loop that I explained before lies in the substrate binding site of CDK, and the phosphorylated tyrosine prevents ATP from binding to the ATP binding site. And as a result, the CDK cyclin complex is inactive. When threonine 160 in the T loop is phosphorylated, the loop moves out of the substrate binding site, and when tyrosine 15 is dephosphorylated, ATP can bind to cyclin-dependent kinase. These two changes activate the cyclin CDP complex manifold. Further activation is initiated and achieved when cyclin-dependent kinase also phosphorylates and activates a phosphatase that dephosphorylates tyrosine 15. The active CDK cyclin-dependent kinase is now ready for further cell-mediated signaling, and this can initiate various signaling pathways for cell cycle. Now, the active cyclin CDK complex triggers its own inactivation by the phosphorylation of another protein called DBRP, or destruction box recognizing protein. DBRP and ubiquitin ligase then combines together and attach several molecules of ubiquitin to CDK cyclin. Now, ubiquitin is a protein. When attached to another protein, it's recognized by the degrading machinery of the cell, called as the proteasome. Once cyclin-dependent kinase cyclin complex is ubiquitinated, as shown here, it is targeted for destruction by proteolytic enzyme complex called proteasome, and as a result, cyclin is degraded. Now, this is the cycle by which cyclin-dependent kinases are regulated. Growth factors regulate cyclin-dependent kinase activity by controlling the rate of synthesis of a cyclin or a cyclin-dependent kinase, or both. Extracellular signals such as growth factors or cytokines activate phosphorylation cascade, such as the MAP kinase cascade, which initiates phosphorylation of Jun and Phos genes in the nucleus. Now, this results in the activation of transcription factor E2F. E2F, when activated, initiates the transcription of cyclin and cyclin-dependent kinases, and also enzymes for DNA synthesis. As a result of this, the cell enters the passage from G1 to S phase. And this is how growth factors and cytokines regulate cyclin-dependent kinase activity. And in this case, it controls the synthesis of cyclins and cyclin-dependent kinases via transcriptional regulation through the transcription factor E2F. Cyclin-dependent kinases regulate cell division by phosphorylating critical proteins. Transcription factor E2F promotes transcription of genes for certain enzymes for DNA synthesis that leads to passage from G1 to S phase. The retinoblastoma protein, PRB, can bind E2F, as shown here, thereby inactivating it and preventing transcription of these genes. Phosphorylation of PRB by CDK2, as shown here, prevents this retinoblastoma protein from binding to the transcription factor E2F. So when the retinoblastoma protein gets phosphorylated by the action of CDK2 cyclin E, this becomes inactive, and as a result, passage from G1 to S happens. So this is one of the functions of CDK2 cyclin E complex in initiating the cell cycle from G1 to S phase. Now, let us consider an instance when double strand break happens in DNA. So damage to the cell's DNA triggers a series of events that inactivate CDK2, blocking cell division. How does this happen? When double strand DNA break happens, a protein mRNA activates two different genes, ATM and ATR. ATM and NATR activate the transcription factor P53. Active P53 promotes the synthesis of another protein, P21. P21, so happens, is an inhibitor of the CDK2 cyclin E complex. Now, when CDK2 cyclin E is inhibited, the retinoblastoma protein, PRB, is no longer phosphorylated, and it can interact with E2F and inactivate the transcription factor E2F so that it can no longer initiate transcriptional regulation for the synthesis of enzymes that are essential for DNA synthesis. And hence, cell cycle from G1 to S is halted. Now, when the DNA has been repaired, the inhibition of the CDK2 cyclin A by P21 is relieved, and cell division is initiated again. So this is a classic regulatory mechanism by which the activity of CDK2 cyclin A is controlled by the gene P53 and P21.